Hello, this is our last lecture, lecture 15, and we're going to talk about very interesting agents, prions that are not viruses, they're just protein molecules, and are able to cause infectious diseases in humans as well as in animals. And today I will also review the parasitic diseases. Okay, so what are the prions? Prions are proteins, and this is abbreviation from proteinaceous infectious particles. But these prions, so we have the normal form of the prions as well as diseased ones, and usually prions are found on plasma membrane of the neurons. And these red things, these are cells, nerve cells, and we see prion proteins stained in red. And what is the distinct property of prions? They have ability to propagate, not really propagate, but they can interact with the same similar molecule. So one prion protein that is, for example, diseased one can interact with the normal protein and can modify and manipulate their shape, and the normal one gets the shape and conformation of the diseased protein, becomes misfolded, and then this one interacts with another with the neighboring protein, and like this, many normal prion proteins become abnormal, misfolded, and CNS disease develops. Prions cause the diseases of central nervous system. Um, yeah, in mammals and in humans, the prions are found in CNS, mainly normal ones, and normal prions are denoted as PRPC, while the misfolded ones appear PSC. And what happens, and the function of the prions, normal prions, are not well understood. They can be involved in some ion uptake, transport of some ions, like signal transduction processes, but the function is not very well defined. And when the person develops the prion disease, their brain shrinks. When in the brain many misfolded proteins appear, many prion proteins change their shape, they accumulate in the nervous brain tissue and cause formation of the amyloid plaques, and they have toxic effect on the neighboring cells, and due to death of the neurons through apoptosis mechanism, the holes are left in the brain tissue, and that's why the disorders caused by the prions are called spongiform encephalopathies. And here we have an example of Creutzfeldt-Jacobs disease pathology that we see in brain section. We see these uh, holes, we see amyloid plaques, and you see here that the brain shrinks, so becomes smaller in size. And there can be different types of prions, but the mechanism of pathogenesis is the same. Uh, and some prion diseases have species specificity, but some can have the have crossed the species barrier, and from animals, these prions can be transmitted to humans. And prion diseases can be genetically determined, and they occur in families. There are sporadic forms when, due to mutation in the prion gene, there are some changes in amino acid sequence of the prion protein, and the prion protein becomes misfolded. And there's also infectious form when humans or animals consume a food that contain the diseased prion proteins, and then these prion proteins can interact with the normal ones, cause misfolding and development of the disease. And prions are very resistant to different means of inactivation. They're not killed by boiling, they're not killed by disinfectants like formaldehyde, ethanol, ionizing radiation, deoxycholate. They're not destroyed by proteases, but prions can be sensitive to the strong sterilizing agents like phenol, bleach, ether, strong detergents, and autoclaving. Um, and guanidine thiocyanate is considered as effective, very effective means in decontaminating medical supplies. So, as mentioned, prion diseases are confined to the nervous system, and what we see in pathology is this spongiform changes and the formation of amyloid plaques. And what is interesting, prion disease belong to slow infections because incubation period is very long, can be months to decades. And then the disease starts to progress, and outcome is always fatal. And this progression of the disease can last from weeks to years. And there are known cases of remission or recovery from the prion diseases. And during prion diseases, we don't see any inflammatory response or immune response because our immune system considers prions as our own proteins, and they are not antigenic, and we don't have production of interferons, no B or T cell response, and consequently immunosuppression of the host doesn't have any effect on pathogenesis. Uh, and But some chronic inflammatory diseases can have some effect on prion pathogenesis. Because prions have a tendency to accumulate in organs with chronic lymphocytic inflammation. And let's discuss a few diseases, some diseases. Kuru occurred in tribes of New Guinea, and there was a tradition among these tribes to the cannibalism was common among these tribes, and they had tradition to eat the deceased people. And the brain was eaten, muscles, and and also other parts of the body. So the prions could be transmitted to quite easily to the people, and the characteristic feature of the kuru was ataxia, tremors, infectious problems with coordination, but dementia was not so common. Creutzfeldt-Jacobs disease has worldwide distribution. Uh, Mainly it is sporadic. Incidence is 1 per million. And there was also possibility of iatrogenic transmission through medical instruments. And Creutzfeldt-Jacobs disease was characterized with dementia, ataxia, aphasia, and visual loss. And variant of Creutzfeldt-Jacobs disease is infectious form, and it is transmitted to humans through ingestion of beef. And uh, beef, so cows develop bovine serum encephalopathies. These are prion diseases of the cattle, and if the person consumes the product prepared from this cattle, then the person can be infected. And all cases of variant Creutzfeldt-Jacobs uh, disease have occurred in individuals who traveled or lived in countries with BSE, Borean spongiform encephalopathy outbreaks. And the most well-known example is Great Britain, where in 1990s the outbreak of prion diseases, BSE bovine spongiform encephalopathy disease outbreak, huge outbreak was registered. And treatment, we don't have any specific treatment. Treatment remains supportive. Um, and so, but as mentioned, the outcome can always is death. Okay, now let's move to eukaryotes and parasites. And usually this parasitology part is divided into two sections. And firstly, we discuss protozoan parasites that are unicellular. And then in the second part, we discuss the helminth parasitic worms. So let's cover these parasites briefly and let's finalize our course. So what are the protozoa? They are unicellular eukaryotic microorganisms, and they are considered under kingdom Protista. That includes also unicellular algae, as well, and also some unicellular fungi as well. And the size and shape of protozoans vary, and size can range from 2 micrometers to 5,000 micrometers. 2,000 micrometers is quite small for protozoan because this is the size of bacteria. And protozoans, majority of protozoans are free-living forms. It means that they don't cause disease and they live in the environment without harming the others, in the soils, mainly in the fresh or marine waters or brackish waters. And we have 4 major groups of protozoans. We have amoebas, flagellates, ciliates, and sporozoans, and all these 4 groups contain the parasitic protozoans, and especially rich with parasites are flagellated protozoans, for example, Trichomonas, Trypanosoma, Leishmania, these are called flagellated. To sporozoa belong the parasite that causes malaria. We'll discuss parasites from group of amoeba, and among ciliates we have just one parasite, Valentidium coli, important parasite. So let's start with the first group amoeba and which parasites are found in this. So this is amoeba and the differential interference contrast microscope, and amoeba doesn't have defined form, and it develops pseudopods for movement, and usually it is majority of amoebas are free living. But one of the important representative is entamoeba histolytica that causes dysentery, amoebic dysentery. This is bloody diarrhea. And entamoeba histolytica is the second leading cause of death from parasitic diseases, and it is spread through contaminated water or food, and it resides in the large intestine in humans and some animals, including primates. And entamoeba histolytica has two forms, two stages, like cyst, that is environmental form, and it is used for this is the form stage, transmission stage, while we have also trophozoites. This is the feeding form. Cysts don't feed, but trophozoites feed and grow then and give rise to cysts as well. So, what happens during infectious process, and um, how amoeba causes the damage? So, as mentioned, we ingest the cyst either through contaminated water or food. Then the trophozoites emerge from cyst in the stomach and duodenum, and this metacyst, ingested cyst, divides rapidly and produces four amebulae. So cysts or mature cysts have four nuclei, and one amebulae from each cyst nucleus develops, and then amebulae again divide into the 8 small trophozoites, and then the trophozoites go to cecum, they multiply by binary fission, and they give rise to the population of lumen-dwelling trophozoites. And okay, 90% of infections have are luminal, and trophozoites multiply and feed on bacteria. They also feed on red blood cells, and part of trophozoites insist and are released with the feces. And first, the cyst is uninucleated, has one nucleus, then two, then three, and then four, and this is mature cyst. So how disease develops? Yeah, so when trophozoites grow by the binary fission, divide by binary fission, and the entamoeba histolytica produces the proteolytic enzymes, and these enzymes can digest epithelial cells and can hydrolyze the host tissue, and it leads to development of ulcers, flask-like ulcers in the intestinal wall, and bleeding develops before, and necrosis of the tissues, and this comes out together with feces. These are the trophozoites here. These are molecules. You see the nucleus. And the life cycle is described here. I have already discussed. And what is interesting, so here that besides invading the intestine, large and small intestines, the amoeba can make severe damage to the wall of the intestines, can move to the bloodstream, and can infect distant organs like uh, liver, can affect liver, lungs, can affect brain as well, and the amoeba causes development of abscesses in these organs. Um, so, how can we diagnose this disease? We can check feces for presence of cysts and trophozoites. And fecal smear can be done, PCR, monoclonal antibody test can be done, serology, and ultrasonography, computed tomography, and MRI to detect the abscesses in liver, lung, and brain. And treatment for asymptomatic, there are some cases of asymptomatic infections as well. And in this case, paramomycin antiparasitic medication is used in symptomatic cases, metronidazole. This is the amoeba with the red blood cells inside them. And some free-living amoebas that live in outside in the waters, and they like especially warm and muddy waters, can cause serious diseases. So soil and water living amoebas, and they can cause deadly disease, amoebic meningoencephalitis. And so we have different free-living several species that can cause disease in humans. This is Naegleria fowleri, Acanthamoeba, Balamuthia, and Hartmanella. And Naegleria fowleri is found in Europe and North America to cause amoebic meningoencephalitis. And how this amoeba evade our body. This infection starts when the person is swimming in the warm and muddy water and salt contaminated pools. And Naegleria fowleri enters via the nose and then moves to the brain tissue and forms nests of amoeba in brain tissue, and it leads to hemorrhage and damage. And the outcome is always the death, almost 100%, and it develops within a week. Yeah, you see, person is swimming, and the amoeba enters the body and then moves to the brain. And this is the damage caused by the amoeba. This is formation of the clusters. Yeah, we see the clusters of amoeba formed in the brain tissue, and it is seen also here. And besides Naegleria, also Acanthamoeba, at least 5 species have been identified in human tissues. And how Acanthamoeba enters our body, and they can also affect CNS and also can cause eye infections. And Acanthamoeba enters the CNS through skin damage. So first the skin is damaged. There's a trauma on the skin, and the Acanthamoeba enters the skin and then moves to the CNS. And besides this, we have also eye infections, keratitis, and it is connected with the use of non-sterile lenses. Yeah, when people are not using sterile saline for their lenses, the acanthamoeba can get inside and can cause the infection. And acanthamoeba and balamuthia, they can cause granulomatous disease, and this is infection of the CNS. So first is the acanthamoeba invades the body through skin lesion, and then granulomatous amoebic encephalitis develops. And here we have so granuloma formation, while in case of Naegleria we have meningoencephalitis. And acanthamoeba may insist and invade the tissue, in invaded tissue, while Naegleria does not insist in the it doesn't form cysts in the tissue. And this is the eye damage due to acanthamoeba. We see the opacity of the cornea and can be associated then with some visual disabilities. And about the diagnosis, so we can check several spinal fluid for presence of trophozoites, and amoebas can be also cultured on special culture media containing E. coli as a food. And for treatment, amphotericin B is used, but with a few success cases. So under ciliates, we have, so ciliates are mainly free-living organisms. They have cilia. These are locomotory organs, body parts, hair-like organelles, and they are used for locomotion as well as collection of the food. And Bacillium coli is the ciliate that causes disease in humans. And Bacillium coli is found in animals as well as humans. And the life cycle of Bacillium coli is very similar to the acanthamoeba. It is also released with the feces in the cyst stage, and then the cyst can be ingested by either animal or human, and can invade the intestinal tract and cause damage there and leading to diarrhea. But here symptoms are less severe than in case of entamoeba histolytica, and Bacillium coli does not go to peripheral organs like lungs and liver and brain, and is not forming the abscesses there. And it is mainly zoonotic infection, but people who have contact with the animals, for example, farmers, are under the risk of development of this infection. And treatment can be done through antibiotics like tetracyclines, also metronidazole, and iodoquinol. So let's move to the sporozoans, and to this group belong also Plasmodium that causes malaria. But let's start first with the Cryptosporidium. The Cryptosporidium causes also GI tract infections, and it is mainly transmitted through contaminated water or physical contact. And in healthy people, it is usually short-term infection, short-lasting infection. So the parasite is transmitted through oocysts that we see here, and this oocyst exists in the small intestines and forms trophozoites, and trophozoites multiply and cause damage. And diarrhea can last for 1 to 2 weeks maximum, but immunosuppressed like HIV patients they can develop cholera-like diarrhea. As for other diarrheal diseases, for treatment, we have here fluid rehydration and electrolyte replacement. Toxoplasma. Toxoplasma gondii is an obligate intracellular parasite, so it goes inside the cell and causes disease, toxoplasmosis. It has worldwide distribution and is found in all warm-blooded animals, but cats are considered as definitive hosts, and definitive hosts are the hosts in which parasites undergo sexual reproduction. This is the cycle of toxoplasma. So usually cat is the uh, definitive host. So cat release the cysts with the uh, feces, and the cysts can contaminate the environment, and in the animals, uh, the the cysts exist, and they then move to the blood and can move to the different tissues, and then in the tissues they are turned into the tissue cysts, um, and similarly humans can be affected, yeah, especially the people who have cats and they are cleaning the cat litter. They are under risk. Of course, not all cats have toxoplasma because usually domestic animals undergo the helmintization, and then they get antiparasitic drugs. So humans can get infected through fecalocysts as well as from the tissues cysts, and both cysts turn into the tachyzoids after ingestion, and these tachyzoids move to the CNS or muscle tissue and turn into the tissue cysts, the bodyzoids. And what is important in pregnant women, the infected pregnant women, tracheozoids can infect fetus and can cause damage to the fetus. So, what are the symptoms in healthy people? Body aches, swollen lymph nodes, headache, fever, fatigue. Symptoms in babies born with the infection, seizures, enlarged liver and spleen, jaundice, eye infections, and symptoms in immunosuppressed, of course, are more severe with the involvement of the lungs, uh, eye infections, and so different symptoms are listed here. But majority of infections are asymptomatic. And we have different antiparasitic medications for treatment. And now let's move to the sporozoan, another sporozoan, Plasmodium, that causes malaria. And it is transmitted through mosquitoes, Anopheles mosquitoes, and Plasmodium species are also intracellular parasites, and they can live in red cells and also in the cells of other tissues as well. And malaria is a quite serious disease, can be fatal as well. And maybe you have heard about typical symptoms in people with malaria. They have um, paroxysms, they have fevers, then a remission-like stage, then again fever, shaking, chills. They have flu-like symptoms, can have hepatitis and jaundice as well. And there are four major species of Plasmodium that can infect humans. Plasmodium falciparum, vivax, ovale, and malaria. The Plasmodium falciparum causes more severe disease in general. And annually, there are up to 500 million cases of malaria, and out of these, 1 million people die annually due to malaria. So the sporozoans have complicated life cycle, and they have two hosts and multiple stages. And the infection starts with the bite of the mosquito, and they have two stages. In human body, we have unsexual reproduction, and in mosquito there's a sexual reproduction, and in human we have exoerythrocytic stage when the parasites multiply in the liver, and after that they move to erythrocytic stage. They replicate, they not replicate, but they propagate. They're not viruses. They grow in the red blood cells. And in human body, they form gametocytes that are uptaken by mosquitoes, and there is a completion of gametogony, so two gametocytes fuse, and then we have the sporogony stage as well. This is also uh, a type of sexual reproduction. So we will not, you don't need to know these details, so let's skip this slide and move to signs and symptoms. Incubation period can be up to 30 days, and the disease starts with the flu-like symptoms, and the people have this headache, fever, shivering, joint pain, vomiting, hemolytic anemia, jaundice, hemoglobin in urine, retinal damage, and convulsions. And classic symptom of malaria is paroxysm. This is a cyclical occurrence of sudden coldness, followed by shivering and then fever and sweating. And it can occur every 2 to 3 days in Plasmodium vivax and ovale, and every 4 days in Plasmodium malaria, while Plasmodium falciparum has fever, almost continuous fever, and that's why Plasmodium falciparum has a more severe outcome. And treatment can depend on species. So, for example, Plasmodium vivax can be treated with chloroquine and primaquine, while Plasmodium falciparum can be treated with artemisinin and combination with other anti-malarial medications. And prevention can be achieved through vaccination. A few years ago, I think in 2015, the vaccine was approved against malaria, and it seems to be effective. And this is distribution of malaria disease. So we have mainly tropical and subtropical areas here. Okay, now let's move to parasitic flagellates, and let's start with the flagellate that causes GI tract infection. It is quite common, has worldwide distribution, and this is Giardia lamblia, and it is found in GI tract, duodenum, and jejunum of human. So these parts of GI tract are affected and can be found in animals. And it has the cyst as well as trophozoite, and it is motile, has the very funny appearance with eyes, so it has flagella, and it has also a special disc, and with this disc it attaches to the intestinal wall, intestines of the small to the small intestine wall, and then it interferes with the absorption of the fats and other nutrients. And the transmission occurs through food or contaminated water, where cysts are found. Then trophozoites emerge and they attach to the intestinal lining, and symptoms develop after about 7 days, and people have nausea, cramps, flatulence, diarrhea, malabsorption, yeah. And this disease is very common in children, um, and also it is common in camping areas, and it is also considered as traveler's diarrhea because of poor hygiene conditions. And in the field, the people can quite easily infect it if the water is contaminated, drinking water is contaminated with animal feces, so they can quite easily be infected. So, the diagnostic stage is either cyst or trophozoites that are released with stage. Infective stage is cyst. It is found in feces, but when there's a diarrhea, trophozoites can also be found. And in colon, the cysts are formed, and they are quite resistant to unfavorable conditions. So this is the cycle. Yeah, we consume Giardia lamblia, it goes in the small intestine, forms trophozoites, and while passing the GI tract, it again turns into the cyst and is released with the feces and contaminates the environment. Diagnosis is done based on a microscopic examination of stools, and cysts or trophozoites can be found. Also, ELISA enzyme linked immunosorbent assay can be used to detect parasitic antigens of parasites. String test is used. This is like a jelly capsule attached to the string, and it is swallowed, and when it's swallowed it moves from the stomach to duodenum, and after some time it is taken back, and on the sticky jelly is examined under the microscope, and parasites are visualized. So about epidemiology, I have mentioned this is zoonotic, has worldwide distribution, and is widespread in areas where it's poor hygiene conditions, and in wilderness and campers are at risk because animals can contaminate the drinking water sources like springs. And about treatment, we have different options, antimalarial, antiparasitic medications, and they are quite effective, and usually symptomatic patients are treated. So now the flagellates that causes sexual STIs, sexual transmitted infections, and this is Trichomonas vaginalis. This is, but there are also other Trichomonas hominis, and Tenax that are, yes, don't cause infections. Trichomonas tenax is found in mouse communities as well, while Trichomonas hominis in intestine, and Trichomonas vaginalis is found in urogenital tract of men and women. We don't go into the morphological detail. It has some, of course, the morphology is important for diagnosis. The flagellate has flagella, of course, and this shape, yeah, slightly elongated shape under the microscope. And the Trichomonas, unlike the parasites previously described, parasites doesn't have this stage because it doesn't live in the outside the host, and it has just trophozoid form, and it is transmitted sexually from one person to another. Anticommodus vaginalis is causing inflammation, and there are many asymptomatic infections, and the disease develops when pH level of the vagina goes up, when there's a change, a disbalance in the bacterial flora, and usually organisms don't survive under normal vaginal acidity. And in females, the parasite affects vulva, vagina, cervix, but it doesn't move to uterus. And uh, there is a discharge, specific discharge. Also, there's inflammation, there's itching, burning sensation. And in males, the parasite affects prostate, seminal vesicles, urethra, and about 10% of males can have urethral discharge. And diagnosis is done by examination of the discharge, some swabs from the discharge, dried smears, yeah, and stained, then that are stained then, and the parasite is detected under the microscope. And the Heterocomonus can be cultured as well and can be then identified. For treatment, we use metronidazole, tinidazole, and of course, both sexual partners should be treated, even if one of the partners is asymptomatic, because there are many asymptomatic infections. So it is very common parasitic disease, and so many so around 50 or 40% or higher, or even more people can be affected. And the majority of infections are asymptomatic. It is transmitted mainly through sexual intercourse, but can be the assumption that it can be also transmitted through towels, douche equipment, examination instruments, and so on. Hemoflagellates. Hemoflagellates, these are flagellates that affect blood and tissues. And under hemoflagellates, we have two major groups, trypanosoma and leishmania, and they are mainly distributed in tropical and subtropical areas, and trypanosomas are distributed in Africa and America, and African trypanosomiasis or sleeping sickness is transmitted by tsetse fly Glossina, and is caused by Trypanosoma brucei or rhodesians or Trypanosoma brucei gambians, depending on the distribution area. And there's also American form of the trypanosomiasis called also Chagas disease. Chaga was the doctor, physician who described this disease, and it is transmitted by the kissing bug Triatoma cornus bug, and in this case we have another species, Trypanosoma cruzi. Leishmania. Under Leishmania genus, we have many species, and certain species can cause cutaneous leishmaniasis, others mucocutaneous, and some species visceral, the most severe form, leishmaniasis. And leishmania are transmitted by the sandflies. So, Trypanosoma rhodesiense and Gambians, they cause both the American sleeping sickness, and Trypanosoma rhodesiense is distributed in East Africa, in East, and Gambians in West, in Gambia, that's why the name, and both cause the sleeping sickness. Trypanosomes multiply in the peripheral blood, but then they move to the CNS and lymph nodes, both. So let's keep the cycle and move to the disease. The more severe disease is caused by the Trypanosoma rhodesians, while the Trypanosoma gambians has more slow development. And what are the clinical manifestations at the site? So it is transmitted through flight cells, yeah, and at the site of the bite, this providing painful shank, the primary lesion develops. Then the parasite moves to the lymphoid tissue, and then finally parasite invades CNS. Firstly, the person is hyperactive, then this stage is followed by lethargy, tremors, meningoencephalitis, cachexia, what we see here, and um, distinct sign of the disease is Winterbottom sign. This is enlargement of the lymph node in the posterior cervical region that we see here. And in the later stage, the patient falls into the sleep, continuous sleep coma, and dies. And as here we have comparison of clinical features, and we see that rhodesians has more rapid development, and the death occurs within the month. This is Trypanosoma gambians in blood. And diagnosis is done based on a microscopy of the blood, lymph nodes, or shank, while gambians is found in lymph nodes. And all patients should be checked, all patients with African trypanosomia, they should be checked if the parasite has reached the CNS, because then the treatment can be different. So before reaching the CNS and development of encephalitis, we have suramine and pentamidine. And if there is also CNS symptom involvement, we have suramine and also melarsoprol that can far pass the blood-brain barrier. So Trypanosoma cruzi, Chagas disease, transmitted by the redweed bug, kissing bug, and the special stage amastigote enter the bite site, but this bug transmits this parasite through feces. When it bites the person, the pruritus develops. The person starts itching, and while feeding on blood, uh, this bug defecates and releases parasites. And when the person is itching, it scratches the skin, and amastigotes, the form of the parasite or stage, enters. And amastigotes enter the wound, then they infect macrophages, and through macrophages can take the amastigotes to different parts of the body, including heart muscle and CNS. Um, so distribution is in Mexico, Central and South America. This is the cycle. We'll not go into the details. And the symptoms, so we have two stages, acute and chronic phase. And in acute phase, we have Romagna sign. This is the painless periorbital swelling of the eye. And it develops because why the name kissing bug? Let's first determine this. Describe this. Yeah, this bug usually bites on face, and especially the eye area. So as if someone is kissing on the face, yeah, person. And Romania sign is the biting site, uh, and the swelling develops. And also, the person has fever, hepatosplenomegaly, and trichomastigotes are in blood, and this stage lasts for 2 to 8 weeks. And later, the chronic phase can develop, and in this case, we have neurodegeneration, cardiomyopathy that is characterized with heart arrhythmia and blocks, heart enlargement, apical aneurysm, megaesophagus, megacolon. So as there's a nerve degeneration, the nerves cannot um, transmit signals anymore to the muscles of the heart as well as muscles of esophagus or colon, and they become, there's a problem with the muscle contraction, of course, and the megaesophagus and megacolon develops. Yeah? And we have treatment in acute phase, and different stages of the parasite can be killed. And when the chronic form develops, there is no treatment. And the last protozoan to discuss today is Leishmania. And this is a disease of mammals, it is zoonotic disease. Mammals are affected. We have many species, and as mentioned, we have three types of clinical manifestation and disease. Visceral leishmaniasis, most severe, mucocutaneous leishmaniasis, and cutaneous leishmaniasis. Here are examples of some species, but there are much more. Leishmania donovani causes visceral. Leishmania tropica major, Ethiopica mexicana causes cutaneous, while Leishmania brasiliensis causes mucocutaneous leishmaniasis. So let's keep this infectious cycle. Parasite is transmitted through the sandfly bite of the sandfly, and the parasite grows in the macrophages, moves to the lymphatic system, lymph nodes, and blood is and parasite gets into the blood, and then moves to target organs like skin, lymph nodes, spleen, liver, bone marrow. These are promesticus, one stage of the parasite. This is a mastigote. So promastigote is motile form, and a mastigote is a motile form, intracellular form. This is macrophage infected with a mastigotes. So phlebotomine sandflies transmit the disease, and these phlebotomine sandflies are very widely distributed. There are 500 species. And which are the mammalian hosts. So you see different mammals. Important are the dogs, especially stray dogs serve as a good reservoir for leishmania. And visceral leishmania, and the incubation period for visceral leishmaniasis can be several weeks, and person has low-grade fever, hepatosplenomegaly, bone marrow hyperplasia, anemia, leukopenia, cachexia, hypergammaglobulinemia, bleeding from the nose, proteinuria or hematuria. So this is quite severe disease. So this is the boy infected with the parasite and having the visceral leishmaniasis, and we have the abdomen is bigger because of splenomegaly, and the boy has the muscle wasting as well. Cutaneous leishmaniasis is milder form, and in this case skin layers are affected, and these type-specific ulcers develop, and then it can heal after some time. Mucocutaneous leishmaniasis is more severe than the cutaneous form because it moves to a deeper layer, deeper tissues, and can cause destruction of the mucous membranes of the nose and can disrupt also um, tissues of the mouth and throat cavities, and the person becomes disfigured. Treatment. So we have two different treatment options: amphotericin B or sodium thiogluconate, and as a control, vector control, reservoir control, so control of sandflies, control of animal reservoirs. So usually phlebotomycin flies appear in the evenings, in the areas close to the forests. So people, if there is an outbreak or if there's an endemic area, one should avoid walking during this daytime. Okay, so we have finished the parasitic protozoans. Let's move to the parasitic worms or helminths. They are quite scary for many students. And when I start the lecture, I ask these questions to the students, the very simple ones. If the worms are vertebrates or invertebrates, of course they are invertebrates, and they have different sizes. How do we measure them? From micrometers to meters. This is the worm, smallest, one of the smallest worms found in hydrothermal vents. These are the areas like small volcanoes in deep waters of oceans, and they are very diverse in they have high biodiversity, and this worm, smallest worm, is found. This is the largest worm, so measures several meters, and this is a free-living worm in water. And where do the worms live? Many of them are free-living, of course. They live in water, soils, but some can live in the bodies of other animals, including humans. And we have several groups of worms. We have flatworms, roundworms, and segmented worms. And flatworms and roundworms are associated with the diseases in humans. So, helminths, parasitic worms, belong to kingdom Animalia. They are animals. And we have two major phyla that causes diseases in humans: flatworms, Pathelminthes, and we have here two classes: flukes and tapeworms. And another phylum is nematodes, so roundworms. And the parasites have some common characteristics. They have reduced digestive system. So because they usually live in the highly nutritious areas where the food is at least partly digested, they don't need complex digestive system. They have reduced nervous system because they are not predators to chase the prey and to need some very well developed nervous system. They merely just sit in one side and feed. They have reduced locomotions. They don't need to move much within the body, but they have complex reproduction because they have to spread efficiently. Yeah, so that's why the production is very important. Helminths have different eggs, types of eggs, and eggs serve as a way of diagnosis. So eggs can be used for diagnostic purposes, and they have different layers. So the shape, layers, type of layers. Of course, the what is seen in the center. Yeah, everything is taken into consideration. Usually helminths eggs are very hardy in the environment. They have several layers that make them very resistant. And helminths can be either monoecious, hermaphroditic, so male and female reproductive systems are in one animal, or they are dioecious, when males and females are separate organisms. And usually life cycle of helminths is like this: egg, larvae, and then adult. And adults lay the egg, eggs turn into the larvae, and then larvae into the adults. Here we have example of monoecious hermaphroditic flukes. Yeah, they have ovaries, testes, both, and also here this is Clonorchis sinensis, also a fluke, and it has testes and ovaries. Yeah, so both are hermaphroditic. Why should we study helminths? Because they are major cause of morbidity and also mortality, especially in underdeveloped countries. And helminths affect almost a third of the world's population. And the diseases are especially important in children because it can lead to physical, nutritional, and cognitive impairment, and global burden of helminth diseases extends that of conditions such as malaria and tuberculosis. This is the spread of the different helminthic diseases, yeah, hookworms, roundworms, whipworms, and we see that these diseases are mainly widespread in underdeveloped countries and in subtropical areas. And the most which worms are the major cause of the parasitic helminthic diseases. This is Ascaris lumbricoides. This is roundworm, and you see there are 1.8 billion cases, huge number. Hookworms, yeah, 1.2, and then others. The worms have a bad reputation, but there are some theories that have actually laboratory proof or observational proof that, like our microbiota, helminths could be beneficial to their host as well because we have co-evolved together, and the increased incidence in allergic diseases, in autoimmune, so chronic inflammatory diseases, um, can be due to lack of parasites in diseases. This is hygiene theory, and that claims that this hygiene can be at very high level. Yeah, hygiene can be harmful for people, and that helminths were reducing the inflammation in general. They were modifying immune response, and that they were actually could be beneficial for hosts. This is red tapeworm, very funny appearance, and nowadays the different parasites are used not officially, and it is not well established ways, techniques of treatment, but parasites can be used to treat some diseases, chronic inflammatory diseases, and this is example right one of these tapeworms, this red tapeworm that is used for treatment of so number of diseases. So, helminthic diseases can be grouped in two major groups. We have intestinal helminthic diseases and blood and tissue helminthic infections. And I have already described that interdefinitive host is the host where the parasite reproduces, and secondary is the host or intermediate host where we have a sexual reproduction of the parasite. Okay, so let's start with the first group, tapeworms. We're talking about flatworms, yeah, and one group of flatworms are the tapeworms, and they are very long. I will not go in very detail, but they have head, they have neck, they have proglottids, and the worm grows at the neck section when where new proglottids are formed, and the last proglottids contain the eggs. Mature proglottids have many eggs inside. And there are two types of tapeworms, the beef and pork tapeworms, Tenia saginata and Tenia solium. And they are acquired by consuming inadequately cooked beef or pork, and then the head, usually the larva is consumed, and from larva the big worm grows, a head attaches to the intestine, and then the worm grows. So you see, Bihu tapeworm is quite very big, can reach up to 10 meters and can live for 25 years. So it has head and it has proglottids, of course. And infection, a source of the infection is, of course, the um, beef that contains encysted larva, Cysticiarchus, and we ingest, and then from larva the complete worm grows. Humans are the definitive host because humans it's the sexual reproduction, and the worm becomes adult within 3 months. And many of these infections are asymptomatic, but symptoms can be gastrointestinal disturbance. Tenia solium, pork tapeworm. It is smaller than the beef tapeworm. It can reach up to 4 meters. And similarly here, the source is the pork meat that contains raw pork that contains the encysted larva, and then this encysted larva turns into the worm when getting into intestines. And Tenia solium can cause also cysticercosis. This is when the person consumes the eggs as well. So this is the life cycle, and so I have explained. So a person consumes meat contaminated with larva, then from larva the complete adult worm grows, and with the it lays the eggs, and the eggs come out together with feces. Then the eggs can be consumed by the pig, and this egg then uh, turns into the larva and can it burrows the intestinal wall and moves to the blood and can get stuck in different tissues, including muscle, that is then consumed by the human. But if the human gets contaminated egg, yeah, also this from the egg, the larva develops, and it can get stuck in the brain in different tissues. And the disease is cysticercosis. This is cysticercosis in the muscle larva of the tapeworms. So Dipylidium caninum, this is dog and cat tapeworm, and here this is zoonosis, and intermediate host is a flea, and mainly children are infected when they ingest accidentally flea, and then in the intestines large worm develops, and infections can be asymptomatic, or there can be some GI symptoms. Let's keep the cycle. Diphyllobotrium latum, this is the very huge tapeworm, and this is fish tapeworm. The human is a definitive host, and the human gets infected when consuming the fish, undercooked fish, and fish has this larval stage, and from the larva again the big worm develops, and it starts to lay the egg, and you see the parasite that then later undergoes the complex development in water, changing different intermediate hosts. And we have many asymptomatic infections, but can be also GI tract symptoms and also anemia. Dwarf tapeworm, Hymenolepis nana. This doesn't have the intermediate host, so from the egg develops the larva, and then from larva the worm, and the worm lays the egg, and the cycle continues. It is very common among, can be common among children, and it is small. That's why dwarf tapeworm, 40 millimeters long. Yeah, and also here we have GI symptoms. Echinococcus, this is also zoonotic. It is not a very long tapeworm. It has just 6 proglottids, and dog is an intermediate host, and definitive host, sorry, and intermediate hosts are these sheep, goats, swine, but accidentally humans can become the intermediate host, and what is important here, in intermediate host, the parasite um, forms the big cyst, hepatic cyst, and it can be found in liver or lungs, in the brain, and can cause serious problems. And damage is done to the different tissues. This is hydatid cyst removed uh, by surgery, and you see how big it can be. And in humans, that can be found in, if it's found in liver, there can be GI tract symptoms, nausea, vomiting, abdominal pain. If it's found in the lung, there can be chronic cough and shortness of breath, and it can affect also CNS, and there can be also CNS symptoms, and the treatment is the surgical removal. So in general, diagnosis of tapeworms is done by testing the stool sample, also a blood test for checking for the specific antibodies. Also, if there is a cysticercosis or hydatid cyst disease, the imaging is used. And in general, for treatment, antiparasitic drugs are used that are listed here. And the treatment for invasive infections like cysticercosis or hydatid cyst includes antihelminthic drugs, anti-inflammatory drugs, anti-epileptic therapy, because the CNS can be involved, shunt placement to drain the fluid, and surgery to remove the existing cysts. Trematodes, these are flukes, also these are the flatworms, belong to the phylum Plathelminthes. So we'll not go into the details. They are just they are hermaphroditic. They have both ovaries and testes, and they have a very complex cycle. and can infect the liver flukes the cycle is shown here, and the definitive host is the human, and the human releases the eggs, and then we have several intermediate, several stages, and snail is an intermediate host here, and the larval stage infects human, and the larval stage then moves to the blood and to the liver, and the adult worm lives in the liver, and here we have symptoms. Pain in the area where liver is located, hepatomegaly, but most infections are asymptomatic. There's also lung fluke that affects instead of liver, lungs, and also it has several stages outside the definitive host affecting crabs, the snails, crabs, and humans eat the crab meat, and then flukes larvae go from GI tract to blood and lungs, and symptoms can be coughing here. Blood flukes schistosoma. Uh, so majority of flukes are hermaphroditic, but schistosoma are exceptions. Uh, they show sexual dimorphism. We have separate male and female schistosomas, and we have different species. Schistosoma mansoni and japonicum affect GI tract, while schistosoma hematobium affects the urinary tract, and a person gets infected while swimming or being in the water where the larval stage of the schistosoma enters the body through the skin, and then moves to the blood, and through the by means of blood, then it reaches, depending on species, either blood vessels of the urinary tract or blood vessels of the liver, and uh, male and female flukes live together. Male fluke has this groove, and the female lives in this groove, lies the egg, and the male fertilizes the eggs, and the eggs are released either with urine or with feces, depending on the species. It can lead to quite serious disorders. Many patients are asymptomatic, but long infections, chronic infections, can lead to symptoms. And eggs, parasitic eggs can get stuck in liver, small blood vessels, or urinary bladder, small blood vessels, and can lead to a granuloma formation. And in acute schistosomiasis, the symptoms can be fever, enlargement of the tenderness of the liver, eosinophilia, dysentery, bloody diarrhea. And in chronic schistosomiasis, so we have um, fibrosis and fibroobstructive lesion around the portal vessels, can be problems with the blood circulation and edema. This is the example when there is a bladder or fluid accumulation due to portal vessel blockage. There can be also rash on the skin, and rash can develop when the larvae enters the burrows the skin and enters the body. And at late stage, we have giant spleen, ascites, yeah, what I have mentioned, hypertension of the portal venous system. Our diagnosis is based on detection of ova. Different species have different ova of different morphology, and treatment is quasi-quantum. So let's move to the nematodes, roundworms, and they're quite diverse, many species, and many are parasitic. Trichinella spiralis affects mainly the muscles, are not very big. We have separate male and female worms. and adults are parasiting in the small intestines of the hosts, and they have worldwide distribution. And this is the life cycle. Yeah, humans get infected through consumption of undercooked pork that contains the cyst. But in the pork, in the pig, the pig consumes the eggs, and from eggs the larva hatches and then moves to the muscles and forms these capsules that is consumed by the uh, humans. And in humans, the adult worms develop, and they produce larvae um, that can then go to the blood and go to the muscle and insist there. And Trichinella spiralis can cause muscle stiffness, pain, soreness, skin rash, swelling of the eyelids, and pain. And many infections can be asymptomatic, and it can be prevented by cooking, proper cooking of the meat, or freezing can also kill the parasites. Yeah, so hookworms are interesting parasites. Why? Because they can, if a person walks bare feet, these hookworms can burrow the skin, get into the blood, and then into the lungs, and from the lungs to the GI tract, um, and can lead to anemia, malabsorption, iron deficiency. Hookworms have hooks through which they attach to the intestines, and they can cause bleeding and blood loss and anemia. And this is the hookworm and how it is attached to intestinal mucosa. So the eggs are released with the feces, and in the soil, several larvae. Um, so we have different stages of larval development, and you see the larva can enter the skin and blood vessels, move to the lung, and then from the lung, it is coughed up and swallowed, and it moves to the GI tract and attaches there and feeds. Ascaris lumbricoides is the most widespread roundworm, and the adult worms, male or female, live in the intestines, and infection is transmitted through fecal-oral route, and humans ingest the eggs. Then the eggs hatch in the duodenum, and then the hatching larva moves to the blood, then uh, to the lung, and then this larva is coughed up again and swallowed, and the parasite enters the gut. Okay, and the adult worms develop, yeah, and it can lead to severe allergic respiratory signs and symptoms due to damage caused to the lungs, due to this high numbers of eosinophils and inflammation, can lead to obstruction of the gut segment, can lead to perforation of the appendix or bowel duct as well. This is the life cycle. I will not repeat. And Herbius vermicularis, this is a very small roundworm, and it is transmitted through hand to mouth. Very often we have hand to mouth transmission. From the eggs, the female and male worms hatch, and they live in the ileocecal region of the intestine, and they can wander through the gastrointestinal tract, and at the night time they can come out of the anus to lay eggs on perianal skin, and it leads to itching in the burning and itching in the perianal area. Very often children are infected and they start to itch the area, and again, they can reinfect themselves. Diagnosis is achieved through Scotch tape test that removes eggs from perianal area and can be observed under the microscope. This is the cycle. Yeah, so eggs get into uh, GI tract, and we get adult worms. Adult female comes out in anal area, develops the egg, and there's some scratches, and contaminates the nails and hands, and can contaminate different objects. We have also tissue nematodes. These are GI tract or blood nematodes. We have tissue nematodes, and they can lead to elephantiasis or orthoceriasis. Elephantiasis is caused by Wuchereria bancrofti. It is transmitted by mosquito, and mosquito transmits the larvae, and the larvae enters the lymphatic system. In lymphatic system, the adult worms develop, and adult worms can block the lymphatic system and lead to accumulation of the fluid and swelling. And of course, the larvae can circulate in the blood and can be again taken up by mosquitoes. And treatment is anti-parasitic drug. Onchocerciasis is caused by Onchocerca volvulus. They are transmitted by biting flies, and they introduce into the blood larval stage, and then the larva matures in adults, and they move into conjunctiva. They mature in the conjunctiva, and they cause inflammation, swelling, and can lead to blindness, and this is called river blindness. And treatment is antiparasitic drug ivermectin. This is the life cycle and treatment. Yeah, so I have mentioned already. Trichiura, this is whipworm. It has the whip, and adult lives in cecum and appendix, and humans ingest eggs, and eggs hatch in the intestines, attach to mucosa, and feed on red blood cells, and can cause damage to the GI tract, and eggs are released in stools. And pathology is trichuriasis, anemia, and iron deficiency. This is the life cycle. We have antiparasitic medications here as well. Okay, thank you for attention. With this lecture, I am closing and finishing our course. Thank you for your involvement, especially in the first part of the lectures. You were and tutorials, you were actively involved, but later, of course, it's summer, everyone is tired, you have exams. I'm sure you will pass the final exam successfully, and I will wish you all the best and good luck in your lives. Okay, bye bye.